Okay, well, good afternoon, everyone. This is Jonathan Maudsley. I'm the Science Advisor at the Association of Fish and Wildlife Agencies in Washington, D.C. We're very pleased to be able to present with our partners at the U.S. Forest Service Research and Development Branch a webinar this afternoon. This is part of an ongoing series between the Association of Fish and Wildlife Agencies and U.S. Forest Service Research and Development. Our webinar this afternoon is going to investigate a citizen science team that's been working to monitor a deadly fungus that threatens amphibian communities in Northern California. And our speaker this afternoon will be Dr. Karen Pope from the Pacific Southwest Research Station at U.S. Forest Service R&D. But before we get started, I just want to go over some quick housekeeping details. First of all, we ask that if you are not the person speaking and you have the capacity to do so, kindly mute your phone so that we don't hear extraneous noise on the line. If noise gets too excessive, I will mute all the phones and then bring up the speaker. But for now, let's just, if possible, please mute your phone so that we don't get extraneous noise on the line. Second of all, the webinar today is being recorded, and it is available, will be available at shortly after the webinar at the Association of Fish and Wildlife Agencies YouTube page. If you go to YouTube and type in AFWA or Association of Fish and Wildlife Agencies, you can find our page. It contains a recording of this webinar as well as a number of other recent webinars in this series and other science-related webinars that the association has sponsored with other partners. So we're really pleased to be able to bring this webinar to you today, and a key part of the partnership involves our partners at the U.S. Forest Service. And here to speak to that partnership and to introduce today's speaker is John Roethlisberger, Dr. John Roethlisberger from U.S. Forest Service. John.
Thanks, Jonathan, and hello to everyone. Thank you for joining the webinar today. As Jonathan said, my name is John Roethlisberger. I am the National Program Leader for Fish and Aquatic Ecology Research for the U.S. Forest Service, and we really appreciate the opportunity to partner with AFWA for this webinar series. The purpose of the series is to share scientific research and synthesis from Forest Service research and development research biologists that's relevant to fish and wildlife policy and management. And that is certainly the case today. Today's speaker is Dr. Karen Pope. Karen is a wildlife biologist in the Aquatic Biodiversity Research Group at the U.S. Forest Service Pacific Southwest Research Station. She is stationed in Northern California in Humboldt County, and Karen specializes in applied research directed toward the conservation of amphibians on public lands. Some of her current research involves the effects of trespass cannabis grows on headwater stream communities. She's also looking at the effectiveness of meadow restoration projects in improving conditions for aquatic biodiversity. And she's also interested in and working on how amphibian population dynamics are affected by pathogens. Today she has a little bit of a different twist on the pathogen research. She's going to be talking about a local citizen science project that she's been involved with since 2013 that's been doing some innovative monitoring for amphibian pathogens in her local area. She'll be sharing information from that project as well as lessons learned about how to develop and put together and actually implement a successful citizen science project and make it a good experience for the citizens that are participating. So now I will turn the floor over to her. Karen.
All right, good afternoon. Good morning to the West Coast. First, thanks to John and Jonathan for hosting this webinar, and also to Monica Thomasy and Nicole Zimmerman for inviting me to present. And next slide. First, I will explain a little bit about my research and how it led me to this project. Then I will introduce you to the McKinleyville Bilingual EcoClub. I will explain how the EcoClub got involved in amphibian disease research and why the topic makes sense for a citizen science group. I will cover the role of the volunteers and the scientists involved. Then I'll describe the specific goals of the project and how it was implemented. I'll give my perspective on why the project was successful and the lessons that we learned. The talk will interweave science and citizen science because in reality, one was never separate from the other. All right, next slide. When the global chytrid crisis hit in the early 2000s, amphibian researchers couldn't help but incorporate chytrid in their research questions. So on the right, in Central America, Karen Lips and others traced a path of destruction when the disease moved through the rainforest frog populations. More close to home, on the left, we saw populations of Sierra Nevada yellow-legged frogs plummet when BD showed up in high elevation lake basin in the early 2000s. All right, next slide. So who's the cause of this disease? The Chytridium dendrobatidis is a fungal pathogen that has caused this epidemic in amphibians worldwide. Its life history is important because it sheds some light on transmission among animals and populations, and why some species are more affected than others. BD has a flagellated zoospore that travels short distances in water to embed in the skin of its amphibian host. And it develops into a sporangia where new zoospores develop in the skin of that host. Currently, we do not know of a dormant fifth stage, which is a good thing. When the zoosporangia reach high densities in the skin of a frog, they affect the animal's ability to breathe, hydrate, and osmoregulate, and can lead to death. All right, next slide. So, my frame of reference is that understanding this disease in populations is complicated. So let me go through this graph. This is a stacked graph representing 3 separate populations of Cascades frogs in the Lassen area of California. The x-axis is time from 2008 to 2015, and the y-axis is BD zoospore equivalents, or a measure of infection intensity. The dots are individual frogs, and the lines link the individuals over time. The important thing to see here is that the lower 2 populations crashed in 2013 after a year with exceptionally high infection intensity. We are trying to understand why one population remained resilient while 2 crashed, and what causes spikes in infection intensity. This busy graph highlights the complexity of what is. Next slide. One thing we have noted is that the prevalence of the disease differs over time and among life stages, and it appears that climate and temperature patterns influence disease susceptibility. In this graph, we separated out life stages of Cascades frogs into adults and 2 juvenile classes, and looking at BD prevalence, which is the percent or proportion of animals that test positive for BD. This is over an active season from spring through fall. And what we see is that adults seem to be able to remove the BD from their skins in the warm summer months, whereas subadults do not. All right, next slide. All right, luckily, you are now saved from obscure data analyses that provide more questions than answers. For I'd like to introduce you to this exceptional EcoClub group that decided they wanted to help us understand what was happening with chytrid in our local amphibian community. This group is led by Dr. Rick Botzler, the inspiration behind this project, and an emeritus wildlife disease professor from Humboldt State University, circled in red. Next slide. All right, let's look at the map first. So just in case you aren't familiar with McKinleyville, California, represented by the yellow star on the map, Sacramento is at the bottom center of the map. From there, pack your snacks and Dramamine and head 6 hours northwest behind the Redwood Curtain to the mouth of the mighty Mad River. The EcoClub is not the richest of school districts, is not in the richest of school districts. It is fairly well represented by different heritages, and clearly the club does not age discriminate. While the EcoClub itself is for youth, this project included the kids and their families and friends. Next slide. In the club's initial research, they discovered that our cool, damp climate in coastal Humboldt County supports a rich diversity of amphibians, and that while chytridiomycosis has been found to cause severe declines worldwide and specifically in California, not much is known about the status and prevalence of the disease in our own backyard. Next slide. The research that does exist for Humboldt County includes 2 master species by Nieto and Son, about a decade apart, that found BD in northern red-legged frogs at the Redwood National and State Parks, represented by the blue arrow north of McKinleyville, and 2 additional incidental detections of BD from Humboldt Bay Wildlife Refuge to the south of McKinleyville, also in northern red-legged frogs. Please note that Redwood National and State Parks and the Humboldt Bay Wildlife Refuge were used as our primary sampling locations for the EcoClub study. Next slide. Some of you are probably wondering if setting kids out to find a deadly wildlife disease is a good idea. Hopefully here, and when I review the methods, I will ease your concerns a bit. So why is this topic okay? Some of the reasons are that amphibians are easy, safe, and actually fun to catch. Testing for BD basically involves rubbing a sterile Q-tip 5 times on each thigh, foot, and belly of the amphibian. In addition, we can't catch BD, and we can follow protocols to prevent spread between animals and populations. Remember, it's an aquatic zoospore stage, and it doesn't have a cyst. All right, next slide. So I just want to go over the roles of the citizens and the scientists involved in the project. First and foremost was the EcoClub, who came up with the topic, conducted the study, and participated in outreach activities. The club leader, Dr. Rick Botzler, was in charge of coordination, consistency, inspiration. He was the data manager. He was in charge of quality control and product preparation. On our end, we helped with study design, training, obtaining permits. We helped with implementation and conducted the data analysis and manuscript preparation. The California Department of Fish and Wildlife Service helped with obtaining the scientific collecting permit, processing the scientific collecting permit at a timely manner, and in project support. The state parks helped with site selection, implementation, and an educational product called Parks Online Resources for Teachers and Students, or the PORTS program. The Integral Ecological Research Center and UC Davis helped with sample processing and the quantitative PCR. And the National Park Service helped with site selection and access. And not on this list is the U.S. Fish and Wildlife Service that manages the Humboldt Bay Wildlife Refuge, and they help with site selection and sampling support. Next slide. The project had 2 primary objectives. The first was to understand the status and distribution of BD within amphibian communities on our local public lands. And the second was to evaluate if citizen science with children could provide viable research on this significant wildlife disease problem. Next slide. So we did go through fairly rigorous training. some directed toward the younger members of the team and some directed towards the older members. We hosted a 2-hour-long training session where we discussed the life histories and characteristics of our amphibian taxa and provided information about current knowledge of BD. We discussed basic animal care, highlighting the sensitivity of amphibian skin to toxicants such as sunscreen, bug spray, and soap. Next slide. We maintained health and safety rules to prevent the spread of BD and keep kids from tasting toad slime and foaming at the mouth. Given that it was infeasible for the kids to wear a new pair of sterile gloves for handling each amphibian, we were rigorous about washing hands between handling each amphibian, and key to that was drying hands and keeping them dry for at least 6 minutes. Everyone was reminded of water safety before heading out on each sampling session, and we made sure there were plenty of adults around to slip first on the unstable and slippery ground. I will say spare clothes did come in handy on a few occasions. Next slide. We carefully disinfected our gear and ourselves before and after each survey to prevent transfer of BD or other pathogens between sites. Next slide. And we followed standard sampling techniques for swabbing the frogs and salamanders, weighing and measuring the animal, and labeling samples for processing. Next slide. We took the swabs to 2 excellent wildlife disease scientists, Drs. Greta Wengert and Murad Gabriel, at the nonprofit Integral Ecology Research Center in the neighboring town of Blue Lake. They showed the club how to prepare the swabs for quantitative polymerase chain reaction, or qPCR, to quantify the amplification of BD DNA molecules, if present in the sample. The prepared samples were then sent to Dr. Janet Foley's Wildlife Disease Lab at UC Davis for the actual qPCR. Next slide. Before getting to the results, we checked with the club for some a priori hypotheses. They resoundingly predicted that frogs would have the highest prevalence of BD than salamanders or toads. The ND in the purple represents those that thought there would be no difference among taxa. The club also expected about 20% prevalence overall, prevalence of BD in the samples. Next slide. And the results supported the hypotheses. We found that 23% of all of the 213 swabs tested positive. 23% of the 213 swabs tested positive for BD, with frogs having the highest prevalence. We also saw differences among species, with the foothill yellow-legged frog having the highest percent positive, and we did not find any BD on any of the salamander species. Note that in this graph, no post-metamorphic animals, no larval animals are included, and we did not find BD in any of the larvae. Next slide. Given previous work, we did realize that survey timing and site might also influence prevalence, and we did want to test if there was a difference in results due to a sampler age bias. So we ran some generalized linear mixed models to see if season, species, and age of sampler influence the probability of animals testing positive for BD, while we also controlled for site-level patterns. We found that season, species, and to a lesser degree, sampler, all had an influence on the prevalence of BD in the samples. Next slide. The age of the swabber was the weakest predictor of BD prevalence. We found that children had a higher probability than adults of getting a positive sample. Some would presume it's because of false positives due to dirty hands touching swab tips but I actually think it's because the kids receive the most oversight and we're extremely careful and thorough with their swabbing. In general, we have come to believe that we have a bigger problem with false negatives than with false positives. Next slide. So this might be a little late in the talk for another data graph, but I think these are pretty cool findings that put the EcoClub study right in the middle of what other researchers are finding. So on the x-axis, we have 4 species of frog. The initials represent the species name. So I'll just translate that for you. So on the left is the western toad. In the green is the Pacific chorus frog. Next is the northern red-legged frog, and then the foothill yellow-legged frog. And then on the y-axis is the probability of testing positive for BD. And the 2 main points I want to make here is if we look first at the green, the Pacific chorus frog, we see that regardless of season, so the first green dot is summer or fall, and the second is winter spring, and regardless of season, the probability of testing positive for BD is low. This puts this study in with other studies that presume that the Pacific chorus frog is not very susceptible to the disease and may actually be a reservoir transferring the disease to other populations. In addition, the other 3 species show pretty dramatic but consistent differences between seasons, with low prevalence in the dry summer-fall periods and high prevalence in the cooler and wetter winter-spring. Again, this result supports other studies that have found that BD grows better on amphibians in cool, moist conditions when the animal's innate immune response may be slower than when it is warm outside. Right, next slide. What we learned, I'd say quite a bit actually. First, anurans, or frogs and toads, all tested positive, while the salamanders did not. We had 3 new reports of BD for foothill yellow-legged frog, Pacific chorus frog, and western toad in Humboldt County. We saw differences in susceptibility between anurans and salamanders and between species of anurans, with the Pacific chorus frog having the lowest prevalence. We also saw strong seasonal differences in prevalence for the 3 potentially susceptible species. Finally, trained children, of course with well-trained adults, did a great job catching and swabbing amphibians. Next slide.
Yeah.
Additional outcomes included the youth leader, Dakota Anderson Spirit, presented a poster about the study at the annual Wildlife Disease Association meeting in New Mexico. In addition, the club participated in the park's online resources for teachers and students, whereby the kids were filmed catching and swabbing amphibians and interviewed about the study for a short educational series that was shared nationwide. Leader Rick Botzler and 2 of the children also presented the study at Humboldt State University's Ecology seminar series. Next slide. Oh. And finally, the EcoClub is a lead author on a scientific publication in the Journal of Wildlife Diseases. This paper highlights the results that I've just shared with you and discusses the value of using citizens to conduct the research. Next slide. Michelle Tam has recently showed me a brief on the Forest Service's new Citizen Science Action Plan. While the EcoClub may not have known what we were doing in terms of planning a citizen science project ahead of time, I think we did a pretty good job of meeting the 5 primary Forest Service priorities by developing a project with a genuine scientific goal, providing meaningful public engagement, proactively involving youth, minority, and low-income communities, establishing and sustaining a strong partnership, and managing for data quality. I'd also like to add the importance of translating the data to meet that scientific goal. Next slide. I'd also like to share why I felt we were successful. First and foremost was Dr. Rick Boxer's dedication to the EcoClub and the success of the project. His hard work instilled commitment in the rest of us. We had a sense of purpose, and so it was easy to give regular positive feedback. We set an appropriate timeline for data collection, and we followed with rapid results. We actually followed through with products, including articles in the paper, journal articles, online resources, and presentations at scientific conferences. And of course, we had fun. Next slide. Some final thoughts on targeted citizen science and research. I believe a strong partnership is mandatory, and we must be willing to volunteer time and participate outside of work. It's important to instill a sense of curiosity and purpose and believe in the value of the project. In truth, we can't instill in others a sense of purpose without that belief in the value of the project. Next slide. And with that, I'd like to acknowledge the stars of this show, both the youth and their family and friends that participated in data collection and design of the project. I'm not going to read the names, though. All right, next slide. I'd like to also acknowledge the multi-organizational partnership that supported and helped with the project. You can see how many and how diverse they are. Next slide. And finally, the ever-important financial support for the project. I'd like to note that most of these are local organizations or individuals, and that bake sales and car washes were involved as well. And with that, I'd like to open it up for questions.
The conference has been unmuted.
Great. Thank you very much, Karen, for that presentation. We did have one question that was submitted using the chat box in the lower left-hand corner of the screen. That's from Bruce Marco, who asks if you could possibly hypothesize why the salamander species might have been BD-free.
Sure. One is that, in general, the particular species we were looking at are less susceptible. The other is that they have a terrestrial life stage that probably allows them to fight off BD because the zoospores can't spread through the water. So densities will be lower on the skin, and then the animals will be able to get through the BD process and shed the disease. There's a lot of research on it, and we frankly don't know how susceptible a lot of individual species are. It just isn't the research.
Very good. Thank you. I see that Michelle Tamez has her hand raised. Michelle, do you have a question?
Yeah, thank you. Thank you for this awesome presentation and for walking us through not only the research but also, how you established it with your citizen science groups and all the people that were involved to make it happen. I'm wondering, because the work was done outside of the National Forest System, if it's something that anybody is thinking about applying on the National Forest. And I hope you're connected with, I think it's Dee Olson who does amphibian surveys. And so maybe there's an opportunity to sort of bring that to that area. So are you guys talking about pursuing this further, or was it kind of a one-time thing?
At the moment, it was a one-time thing. I think it can serve as a platform to do additional work like this elsewhere and on Forest Service lands. Dee Dee and I communicate. We haven't set it up. We're not in the same areas, but we have suggested that we do this work in some of the areas. We work in the Latin area, working with the school districts out there to set up a educational program that incorporates the schools, the after-school activities, and our research in meadow systems.
And so you mentioned talked a little bit about whether citizen science was a good, or, whether the science that came from the citizen scientists were good. And I'm wondering, did you have other evaluation parts that you were looking at besides just data quality? Like, I don't know, for instance, how much the kids learned, or any other kind of evaluation criteria for the project?
Yeah, so the Humboldt State University participated, and they designed a test to inform us of what the kids learned from the beginning of the project to the end, and I actually have not seen those results. We did try to implement that to understand how much education there was.
Okay,
but
cool.
I don't know the results.
Okay,
Great.
well, I would love to follow up with you on some of that stuff. So thank you again.
Great,
Sure,
thank you. Great questions. We've got a couple more things here in the chat box. We have one from Alyssa Wetterow asking if field survey protocols were optimized for capturing terrestrial salamanders.
We focused on aquatic habitats, but caught some animals in the near terrestrial areas. Frankly, the kids were looking in places that a lot of us wouldn't have looked. We had a kid climbing a tree, and I told her to look around while she was up there, and of course she caught a couple Pacific horse frogs hiding under a piece of bark. So we did have some that were not in water at the time of capture, and we just incorporated them in. It was noted that they were found on land, but we didn't have different directed survey protocols for water or land.
Great, thank you. Question here from Chelsea Clockey. Are there plans to do future monitoring with the club or another project?
Right now the club is looking into starting a project up in Redwood State and National Parks, looking at elk movement, and I am not involved in that part of the project, but they are looking to try something else.
Great. Question from Simon Ray. What was the ratio of adults to kids in the field?
It was usually close to 50-50. We never went out with any more than 2 to 1, 2 kids to 1 adult. And it was usually at least as many adults as kids.
Great, thank you. At this point, we've run out of questions in the chat box and also hands raised. So we do have some time left. We usually try to allot up to an hour for these webinars. So if there are any additional questions for Karen, we'd certainly invite you to just go ahead and speak. The lines have been unmuted, so please feel free to ask Karen any questions you might have, and we'll keep the line open as long as we need to address all the questions. Thank you.
Hi, Karen. This is John Roethlisberger. Thanks for your presentation and great project. Was curious if there were any extra challenges or questions that were asked when you went to obtain the collection permit that you needed, given the involvement of the club?
Yeah, not really. Both Rick Botzler and I have a history of having scientific collecting permits, so it was fairly easy to add this project to our existing collecting permits, and the state was really supportive of the project, so they actually moved it through fairly quickly, and so that we were able to get the permit when the kids were ready to get in the field. So it was a fairly smooth process. We also had an ISO through HSU, through Humboldt State.
Well, that was actually, this is Michelle again. I actually had that question too, but I was kind of wondering if maybe that would be a reason why this type of work wouldn't happen on a national forest, is how difficult it is to get permits or to actually have the kids go out there. Is that something that you've faced?
I think if the right people are involved with the skills, that the permits would not be a problem. Certainly if it was in the Sierra Nevada dealing with listed species, then there might be a problem. But if we focus on more common and local taxa, I imagine that the permit process would also go smoothly.
Any other questions for Karen?
This is John. Yes, I've got another one. How did you...
Hello.
Oh,
Karen, this is Bruce Marco. I'm sorry, can you hear me?
Yes. Hi, Bruce.
Okay, hi. Way back in the 70s, I did studies up on Six Rivers National Forest on pond habitats, in which I did surveys of perps, including frogs. And I'm wondering if BD tends to be less transmissible within or among pond or still water situations than in streams.
Yeah, I think that's somewhat of an unknown. There's been some work in Central America that's shown that stream amphibians were more likely to have BD, and then in our work here in the mountains of California, we find that BD reaches higher densities in still water habitats, and that when the animals move into streams, it seems like they have a higher likelihood of becoming BD negative. So it kind of makes sense that within ponds where densities of animals can get to high numbers, and then also where the zoospores can move among animals, it can reach some high densities. The water can get pretty warm, and I think that allows frogs to fight off the BD as well, though. So it's a mixed bag, and I don't think we fully understand the complete environmental connection with the disease host dynamics.
Thank you.
John, did you have a question?
I did. I had another couple of questions. One was, could you talk a little bit more about how you got connected with the club? And also, a second question is, how did you choose sampling sites?
Yeah. Okay, great. So how I got involved in the club, Rick Botzler lives down the street from me and knew that I was doing work with amphibians and BD. And so he approached me and said that the club was interested in doing this type of work and what I thought about it, and would I be interested in participating and being a part of the study. And so that was basically all it took, and we went from there. I admit that at that beginning stage, I was first a little sheepish that I didn't know much about what was happening in our own backyard. And then, two, I also wasn't sure where the project was going to go, and was really pleased that we were able to just keep it going and collect really good data. In terms of where we decided to go, part of it was based on where the historical sightings of BD were, and then also local, easy to access public lands where there was plenty of aquatic habitats and amphibians. So that pretty much was the deciding factor on where we would go. We did visit some local areas, usually in training when new members joined and such, but the data mostly is from those two public land areas.
Great, thank you, Karen. Yeah, go ahead, please.
This is Michelle again. I'm wondering, you had the slide with all of the organizations that donated money for this study. Could you talk a little bit more about what the cost was and how that money was used for this, and also how those organizations were brought into the mix?
Sure. So the primary costs went to supplies, so the nets and disinfectants and some boots for the kids, and then also for taking Dakota to New Mexico to present the poster. So we sent Dakota and her family to New Mexico. And so that involved a fundraising activity, and I think most of the funds came in through an article in the paper, the local paper, the Times Standard, whereby we solicited it. And then also the kids raised funds through car wash and bake sale. And then I forgot what the second part of the question was.
Yeah, I think that was it. It was kind of, how those organizations got involved and how you did that. So that part about the local newspaper article and funds is an interesting one. And also I thought that was really cute at the end, seeing the car wash and the bake sale, and that's great that, they're raising their own money, that they care enough about it to do some fundraising for it in that way.
Yeah, they're a pretty impressive group, actually.
Any other questions for Karen? If not, I'd really like to thank Karen for the excellent presentation and for everyone who asked questions and contributed to the discussion. This is a very interesting, very informative webinar today. And just to remind everyone, this has been recorded and it will be available on the Association of Fish and Wildlife Agencies YouTube channel shortly. If you go to YouTube and look for Association of Fish and Wildlife Agencies or AFWA, you should be able to find this recording, as well as recordings of other recent webinars that we've offered in this series with the U.S. Forest Service, as well as some of our other partners. So we invite you to go and check those out. And stay tuned. We will be having another webinar in this series next month, and so we will be sending out information about that webinar very shortly. But to wrap up today, I'd like to ask John Roethlisberger see if he has any last-minute and closing comments that he'd like to share with us to close out today's webinar. John.
Sure. Thanks, Jonathan. And on behalf of Forest Service Research and Development, thanks to everyone for participating in the webinar today. I hope that the information that Dr. Pope shared will be useful to you in your own amphibian conservation projects and your own citizen science efforts as well. Please feel free to reach out to Karen or to any other Forest Service scientist if you have questions or require assistance or see opportunities to collaborate on projects related to fish and wildlife conservation that may be of mutual interest. Jonathan and I can help make connections as needed also. And many thanks to Jonathan and to the Association of Fish and Wildlife Agencies for organizing and hosting the webinar today. As Jonathan mentioned, we will have another webinar in this series coming up soon. We're planning for that to be in June. We have not yet picked the exact date, so please watch for more information, which will be coming out soon. We expect that the topic of the next webinar will be the Animal Welfare Act and institutional animal care and use committees for fish and wildlife research. The Forest Service has recently gone through the process of setting up its own institutional animal care and use committee, and our attending veterinarian who leads that committee, Dr. Patrice Klein, will be sharing some insights about that process and just talking more generally about the Animal Welfare Act. So please watch for information about that soon. And more generally, we appreciate feedback about these webinars, and we're always interested in suggestions for topics that would be of interest. So please share those suggestions and share any feedback with Jonathan or myself. And thank you very much. Really appreciate your participation in today's webinar.
Yes, thank you all very much, and hope you all have a wonderful afternoon. Thank you. This concludes today's webinar.